Welcome back, everybody. Today we're going to be talking about mitosis, the process of actual cell division, so creating two new cells. In the last video we went over the cell cycle, so we went over the entire cycle. So we looked at the interphase, which is the longest phase, and the three subphases of G1 being the growth phase, S phase being where that DNA is getting duplicated before cell division. Before you can divide, obviously you need double the amount of DNA. And then that G2 phase where the cell is preparing for cell division. This video we are looking at mitosis, the M phase, which is the actual cell division stages. So once that cell enters the M phase, what does that cell look like as it goes through prophase, metaphase, anaphase, telophase, and finally cytokinesis, where that cell is going to split and create two new daughter cells. So this is the process of going from one cell to two exact identical daughter cells. So mitosis has four phases, and the way to remember the order of them is PMAT. So you have prophase, metaphase, anaphase, and telophase. And for each of these, you need to understand the diagram, so you will be drawing these quite often in class, and you need to be able to, using words, describe to me what's happening in that picture in that phase. The first phase is prophase. Prophase is the actual longest phase of mitosis, which unfortunately means it has the most events occurring during it. So one of the first things that happens is that these centrioles, which we talked about back when we did the cell organelles, we just said that they were used for cell division. Well, now we're actually talking about cell division, so we're not actually going to talk about what those centrioles actually do. So the centrioles during prophase are together originally, but during prophase they separate and move to opposite sides of the nucleus. And as you can see, what they're also doing is they're creating these spindle fibers. They're creating these ropes that we're actually going to use to help lasso the chromosome molecules into line, and we're going to use these ropes, these spindle fibers, to help get the chromosomes into each of the two new cells that we're actually creating. So as I said, you can see that these spindle fibers are forming. Another important factor that happens during prophase is that the chromosomes, the X-shaped duplicated chromosomes, actually become visible. So before this, during interphase, you just see a big pile of DNA. You don't see these individual chromosomes, these X-shaped chromosomes. However, during prophase, the X-shaped chromosomes condense, and they're now visible under a microscope. And as I said, these centrioles, these centrosomes, also known as centrosomes, are going to make these fibers, these spindle fibers, that are going to be used to help get these and rope these chromosomes into line. So the chromosomes are just DNA, so they're just floating around, so we're going to need to help guide them to where they actually need to go. And also, as you can see, if you look at the picture, the nuclear membrane is also starting to disintegrate. So the nucleus is going to disintegrate because if you're separating out and creating two new cells, you have to divide up that DNA, so therefore you have to get to that DNA, so that nucleus has to disappear. And this is basically what I just said. So the DNA, the chromatin, is going to, which is just stringy DNA. So chromatin is just what DNA looks like normally. It's that string form. And we're going to condense that during prophase and create these X-shaped chromosomes. And the spindles have formed, and this nuclear envelope, the nuclear membrane, is now breaking down and disintegrating. So you should copy down all of these diagrams in your notes because you do need to know what they look like, and I'm going to ask you to draw them numerous times. So looking at some actual cell images, we can see in prophase, here's some interphase cells. You can see the nucleus is clearly still defined. You can see that the DNA is in that chromatin state, just that ball and the string state, just stringy DNA everywhere. However, in prophase, you can clearly see now the chromatin is condensing into those actual chromosomes. The second phase is metaphase. Meta means middle. So in metaphase, you can see that these spindle fibers have lined up and have forced these chromosomes into a straight line. So these spindle fibers have attached at the centromeres and have pushed and pulled these chromosomes into a straight single file line directly in the center of the cell. Metaphase, if we look at the actual cell on the right, we can clearly see the DNA is lined up in the middle, the chromosomes are lined up in the middle. We can see the spindle fibers and the centrioles. So it's going to look exactly like the picture pretty much. The third phase is anaphase. You can think of ana as breaking apart. So in anaphase, we are basically draw and quartering these poor chromosomes. So we are ripping each duplicated chromosome into individual chromatids, which now we call chromosomes again. So we literally in anaphase are moving the two identical sister chromatids away from each other, so that each cell is going to get one copy of the chromosome for its use. So remember, each one of these little halves is identical to each other. So this one's going to go into one new cell, and this guy's going to go into the other new cell for its function. So we're separating them into two groups, so we're going to create two identical cells, each with the identical DNA during mitosis. And so anaphase is this splitting event. Anaphase, we can see in anaphase the DNA is now being pulled apart and separated, so the chromosomes are being pulled, the chromatids have been pulled apart. And then the final phase of mitosis is telophase, or telophase. I say telophase, where you can look in the picture, you can see like it's starting to actually pinch off and starting to form two new cells. You can see that the nuclear envelopes, the two new nuclear envelopes are redeveloping, so you're creating two new nucleus, each with the same DNA that started in the original cell. And the chromosomes are also starting to uncondense, so they're starting to unravel back into a string-shaped form, which we call chromatin. And then telophase, we can see that the DNA is starting to compile again, the nucleus is starting to reform, the cleavage is beginning, so cytokinesis is beginning, and we can see that the process of actually separating that DNA has been completed, and the spindle fibers are starting to disappear. And finally, the last part of cell division is not technically part of mitosis. It's called cytokinesis. The movement, the kinesis means movement, cyto means cell, so cell movement, and this is the final pinching event. So if we look back at telophase, we have this cleavage that's starting to occur, so it's starting to pinch in and form a cleavage furlough. And then finally, during cytokinesis, that cytoplasm is going to split and is going to separate, and now we have created two identical daughter cells that are not only identical to each other, but identical to the original parent cell that produced them. So mitosis, the entire point, is to take one parent cell and create two identical daughter cells through prophase, metaphase, anaphase, telophase. Looking at some differences between plants and animals, one thing that you should remember is that plant cells do not have centrioles or the similarly named organelle, the centrosome. So centrioles are not actually required for mitotic cell division. You can get by without them. They don't actually create these spindle fibers directly. They do have a role though in help organizing and pulling apart the mitotic spindles and the chromosomes, but plants obviously can rely without having any kind of centriole whatsoever. If we look at the picture down here, this area where the spindle fibers are created, where the microtubules are created, is what is known as the centrosome. In animal cells, within the centrosome, there are two centrioles. So plants actually do not have centrosomes or centrioles. They do obviously have the proteins inside of them that help create those microtubules to create those spindle fibers, but they are just not organized the same way. So while in an animal cell, we can clearly basically point out and say, this is where the microtubules are originating from, where they're actually being created from. In plant cells, because they don't have to actually physically divide their cells in the same way as we'll see in a second, they don't require like changing the shape of their actual membrane. They don't need to have this organization compared to animal cells. So where the spindle fibers are created, spindle fibers are made of what are called microtubules. These areas, these centrosomes, are what are known as MTOCs, microtubule organizing centers. So they're organizing these microtubules, which are then going to be created and shot out to connect to the chromosomes. So just to reinforce, plants do not have centrioles or centrosomes, but obviously they still are creating microtubules. It's just they don't need the organization that an animal cell does, since a plant cell is not physically splitting its cell membrane or moving its cell membrane the same way that a plant cell is, that an animal cell is, sorry. Animal cells have them, plant cells do not. One big difference also between plants and animals is that in animal cells, they can actually physically pinch in and separate since they only have a cell membrane. Well, plants have a cell wall they also have to worry about. So they don't actually pinch because they have that firm cell wall. Plant cells don't pinch off from each other. What plant cells do is in vesicles, they actually start making their cell wall in between. So you can see they just keep adding materials in vesicles that start combining and create what's known as a cell plate. And eventually that cell plate just keeps elongating and connecting from one edge of the big plant cell to the other edge, eventually ending up creating two new cells. Since plant cells aren't moving apart from each other really, basically you're just creating almost like just building on top of each other, while animal cells physically can pinch off and move away from each other. And just to recap the significance of mitosis, so growth is obviously a very large, important aspect of mitosis, creating more cells. This idea of replacing cells and repairing tissues. So you want to be able to make sure all the cells are identical to each other. Asexual reproduction, so if you're talking about like a yeast cell, it needs to be able to create more copies of itself. And when we get to the immune system, this idea of creating more B cells and T cells for your immune defenses are all created through mitosis.